Good afternoon. Welcome to the University of Melbourne's third MedTech webinar for 2021. My name is Andrea O'Connor. I'm the Shanahan Chair in Frontier Medical Solutions at the University of Melbourne and the Aikenhead Centre for Medical Discovery. And I lead the Tissue Engineering Group in the Department of Biomedical Engineering. I have the pleasure of being your MC for our webinar panel discussion today. And the topic we are discussing today is bioengineering innovations in medtech for eye repair. So I'd like to start today's event by acknowledging the traditional owners of the land from which I'm hosting this webinar, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. The MedTech webinar series has been looking at the key technologies that are being developed at the University of Melbourne by our collaborators and our close friends. And so far, we've been able to see some of the key advances in brain-computer interfaces, regenerative medicine, cardiac technologies, and personalised medical devices. If you'd like to learn more about our research and innovation programs or view any of the earlier webinars, please visit our website. Please note also today this event is being recorded and the video of the webinar will be put on the University's Faculty of Engineering and Information Technology webpage. I'd like to welcome Zara, our illustrator, who will be scribing today's discussion. Hi Zara, thanks for coming. We're looking forward to seeing what you will capture from the discussion today. So a little bit about today's topic. Innovations in medical technology are providing many new ways to prevent, diagnose and treat vision loss and blindness caused by a range of conditions, including glaucoma, diabetic eye disease, age-related macular degeneration or injury. So today's panel brings together some experts in this field. We have clinical, engineering and industry experts here to discuss some of the current clinical and research challenges in this field, as well as some of the bioengineering strategies focused on novel biomaterials that are being developed to tackle vision loss and improve outcomes for patients. So before we proceed with the panel discussion, I'd like to take a moment to thank you for joining us, first of all, and let you know you're welcome to interact by typing questions for our speakers in the Q&A box to your right-hand side. Some of you have already submitted questions via the registration form, but if our conversation sparks further thoughts or questions, I do encourage you to engage with the panel through writing your questions in the Q&A box or by liking the ones that you would most like answered. We'll try to address as many as possible during the session. So to begin the panel, I'd like to introduce our panel members and invite them firstly to give a brief presentation about their experience and knowledge related to technology and bioengineering for eye repair. So first up is Mark. Professor Mark Daniel is an ophthalmic surgeon and head of corneal research at the Centre for Eye Research Australia. Welcome, Mark, over to you.
Thanks, Andrea. Yeah, as you said, I'm an eye surgeon, and my main area of interest is the cornea and doing corneal transplants. If I can go to the slide, I can explain what the cornea is, perhaps. So the cornea is the clear window on the front part of the eye, as you can see on the left-hand side there. And to maintain transparency of the cornea, you need to have a functional layer of cells we call the endothelium, which pumps the cornea dry. As the cornea gets diseased, you lose those cells, and they're not replaced in the body, and so the cornea becomes swollen. And you can see underneath that second image the swelling in the cornea, and it's like looking through frosted glass. Patients do not like being blind, and they will do almost whatever you ask them to do to regain their sight. What we normally do is a corneal transplant, which requires corneal tissue donated by someone who's died, stored in an eye bank, and then distributed, prepared and then distributed to eye surgeons. And that's fine in Melbourne, and it's fine in the US and parts of Europe, but there are many parts of the world where eye banks aren't well organised. And we thought the idea of a tissue-engineered corneal transplant, where you can use one donor and supply many different patients through manufacturing a corneal transplant would be a good idea. We've been working with Greg for the last nearly 10 years now, using a hydrogel that he's developed that has all the requirements we need to carry the endothelial cells into the eye. So the hydrogel is transparent, obviously, it's biocompatible. It's also biodegradable, which means it dissolves without any toxicity to the eye. It's also tough enough for me to operate with. And we've found that using this hydrogel, we can either glue donor corneal tissue to it or grow cells on it. Use that instead of a corneal transplant. So instead of a donor, we can manufacture this tissue-engineered cornea. And with any luck, you end up with a perfectly crystal clear cornea like we see on the right-hand side there. Thanks, Andrea. That's a reasonable introduction.
Fantastic, Mark. It's exciting to see where the research is going, and it really looks like it will have great impact. Next, I'd like to introduce Russell. So Dr. Russell Tate is the CEO of Polyactiva and a co-inventor of Polyactiva's technology. So welcome, Russell. Over to you.
Hi, Andrea. Hi, everybody. Thank you. So maybe if Olivia can put up the first slide, I'll introduce myself. I'm Russell Tate, CEO of Polyactiva. I have a pharm chem background and obviously a business background as well. So Polyactiva is a clinical stage ophthalmology company. We have a couple of assets in clinical development. I'll show a few slides on our core asset. And what we have is a unique delivery technology that is quite unique and very well suited to the eye. So Olivia, if you pass on to the next slide. So what we do is we take existing drugs that have therapy in the eye, we covalently attach them to an incorporating monomer to build up a bespoke drug polymer conjugate is what we would call it. We then formulate that in such a way that it can be administered as an implant through a custom-built syringe, such that once administered to the site of administration, and we can determine which sites in the eye we would like to place the product, it takes up moisture from the surrounding tissue and it relies on the chemistry of the attachment of the drug to the polymer backbone, which is always pendant, to trigger release of the drug. And something's happened with the video there, Olivia. The process then involves release of the drug and then eventually breakdown of the polymer scaffold, so it dissolves and disappears completely in the eye. And with this particular technology, we can control the daily dose that's administered of the drug precisely. We can also control the period over which that treatment is administered. So if we go to the next slide there, Olivia. This slide shows some data for our primary asset, which is an ocular implant for the treatment of glaucoma. It's designed to substitute for daily drops. Glaucoma is a disease of elderly patients. They often struggle to administer drops or forget to administer them. Daily drop administration, particularly for the most commonly used drugs, prostaglandin analogues, can result in some surface eye disease. A lot of these problems are eliminated with a single implant. And so in this clinical study, we've shown a clear reduction in intraocular pressure, which is what these drugs are designed to do to treat glaucoma, at the moment over a period of 18 weeks. And so we're quite excited with this data. Next slide, Olivia. The implant sits inside the eye, so it is administered by injection. It's transparent, you cannot see it. It does not irritate, it's very well tolerated, and importantly, it doesn't move around in the eye, so it doesn't damage that precious endothelial layer that Mark spoke of earlier. Next slide there, Olivia. So yeah, that gives you an introduction to Polyactiva and what we do with our technologies. Thank you.
Thanks, Russell. That's wonderful to see and looks also very promising. Now I'd like to introduce Greg, and Professor Greg Chow is from the Department of Chemical Engineering at the University of Melbourne, where he leads the Polymer Science Group. And so the technologies you've already heard about are based on polymers. And Greg, we'd love to hear a little bit about what you're doing.
Thank you, Andrea. Can we have the slides, please? So my background training is a polymer synthesis. So we train our students to make a polymer in different shape, size, and apply for different application. So this overview shows our work in the group. Top left corner is about how we make polymer in different ways or different environment. The top right corner is where we're looking at the many industrial applications by using those polymers. The bottom right of the corner is where we make a polymer to suit for environmental protection or, for example, saving waters through use those polymers. The bottom left corner is where we're using polymer for tissue engineering purpose. And that's where it's related to the topic we do today. Next slide, please. So if you look at the next slide, the top row is relating to other research we are doing by using amino acids as a precursor to make a polymer. And these amino acids made the polymer may solve fundamental issue of the plastic waste because these are made from amino acids. So therefore, has the same nature cycle for any other material in the nature. We also use those polymers as a drug, for example, to kill the bacteria. And this is a potential future polymeric antibiotics. We also use those material and looking at how we make them as a drug to treat the cancer. But the topic for the focus of today is the hydrogel technology, which is the bottom row, where about 10 years ago, we developed this hydrogel material for 3 main applications. The first one is your bottom left, is where we found use the porous hydrogel can be used as a supporting scaffold to grow tissue. And one of the very important issue is the foreign body responses. We found that the hydrogel compared to other plastic support, we just had so much better results after 8 weeks where there's no foreign body response issue occurred or observed. Mark talked about we worked together for about over 10 years to looking at how use this hydrogel work in the eye repair. So we are working on the 2 projects. One is looking at using hydrogel as a support, which is the blue color there, and we grow the donors tissue together, and then we could use this method to make the surgery easier, better, and also less rejection in the future. And this is a project called CoreGel project. On the bottom right is the one we've been working on for a long time. We're still going on to work this, is looking at how using a polymer as a support and grow the cell in the lab. So the cell with the polymer support itself is the synthetic transplant, and then we can use this to the patient. This will solve a lot of the cornea shortage issue in many countries, and so we are very excited about those 2 projects in the cornea area. Thank you, Andrea.
Thanks, Greg. That's a fantastic overview of some of the very exciting work you're doing and the wonders of polymers and all the different things they can achieve. So now I'd like to bring all our panelists on screen so we can delve deeper into this topic. And I remind you to add your questions in the Q&A box if we've sparked any for you. Perhaps to start off with, I might ask a couple of questions. And if I start with Mark, Mark, are there other unmet clinical challenges that are limiting our ability to prevent or treat vision loss where bioengineering might be able to help?
Well, there certainly are. The way I think about, when I'm seeing patients is about 3/4 of the conditions that I see are either treatable or preventable. And so we've got well worked out systems for treating those sorts of diseases. And the real issue can be with service delivery. In Australia, that means regional and remote areas. Normally the cities are pretty well served. But internationally, there can be supply issues with various drugs, or as we've seen with corneal donors. And so maybe the tissue engineering organs will help with that supply side of things in the rest of the world. The other unmet challenge really is in the diseases that are not treatable and are not treatable or don't have a preventable at the moment. And so there, people are doing a lot of work, especially in degenerative diseases and genetic diseases with gene therapy and stem cell type treatments. And all of these sort of treatments are going to need engineering support. Usually delivering the cells or the drug or the new treatment will require some hydrogel like Greg's making, or some drug delivery device like Russell's working on. And even more technical robotic sort of surgery to get some of the gene therapies have to be placed at exactly the right point within the eye and require a whole lot of engineering to get the surgical or the robotic surgery to get things to that level of precision. So, yeah, I think we're going to have a fruitful collaboration for many years to come as we get all these new treatments going through and being used in humans.
Fantastic. Do you envisage using stem cells with your core gel technology, or would you be using autologous cells?
Well, ideally we would, and I suppose that's our more blue sky type of research. What we're trying to do at the moment is, currently we're using donor tissue and then trying to amplify that in the lab, and then we can use it in many hundreds of cases rather than just one donor per patient. But we're looking at induced pluripotent stem cells and then working out the mechanisms to turn that into corneal cells and into corneal endothelial cells. And obviously that would give you a completely unlimited supply of tissue. And then if we can get them constructed in a way that's safe and effective and we can operate with it, then we'd have an unlimited supply of donor tissue. So that would be really something worth achieving. Might be a couple of years off.
Keep going. That sounds great. Maybe, Russell, if I can turn to you from the manufacturing perspective, what are the main challenges you find in developing and scaling up new products for this kind of application, like an ophthalmology application?
Yeah, I guess there are certainly challenges, particularly for ophthalmology, because there's uniqueness around ophthalmology products because they are administered to the eye. I guess I would say there are two main challenges. One is specialized process developments required to scale up an ophthalmology product. A conventional pharmaceutical product, we tend to focus on the potency so that we get the right unit dose, the purity of the product, so we know what related substances to the drug substance are present and control those. We control unrelated materials associated with the process, like residual solvents, etc. But in ophthalmology, you have additional challenges as well. First, the product's going to be sterile when it's administered to the eye. You can't introduce any bacteria. You've got to control endotoxins like you do for a parenteral product. Importantly, no foreign particles in there, including subvisible particles, and you can imagine the challenges of that with an implant product. And you are required to do 100% visual inspection and make sure that the product is pure. So these are things that are unique to ophthalmology that you have to factor in when you're scaling up your manufacture process and add challenge to that process. And I guess you can imagine with those sorts of features, the other big challenge is the facility. You need a dedicated facility. It has to have a sterile suite. You can't just manufacture this product in any sort of routine environment.
Yeah, that makes great sense. And I guess that leads me to another question. How do you see Australia's capability in that kind of manufacturing, and what sort of challenges do we have in establishing sovereign or local manufacturing for these types of products?
Yeah, the challenge is significant. At the moment, there is no capability in Australia. So we have no established facilities that are dedicated to the manufacture of ocular products. And so we do have to outsource that activity overseas. The barriers to establishing that facility are significant. There's obviously capital barriers, the cost associated with building such a facility. It's often cheaper to engage a facility that exists already overseas, despite the logistical difficulties of dealing with an overseas service provider. There's also expertise barriers. And so finding the talent around to actually manufacture products like this, given it doesn't presently exist, is a challenge. And these are big barriers to entry if we were to develop a facility within Australia as a sovereign manufacturing facility.
Quite a hurdle. Something
Yeah.
for us to keep talking about, I'm sure. Greg, can I turn to you and the polymer science developments that you're leading for applications like ophthalmology? How do you innovate in your polymer science, or how do you think your polymer science research can contribute to meeting these clinical needs?
I think for myself, or even most of the people in my group, we do not have a medical training. So it is actually important to talk to a clinician and understand what the problem is, what is the needs out there. And I think every time I talk to a clinician in different area, I learn lots from them, and often found that the issue is interesting and how we might be able to help. I think equally, you also need to tell the medical society or field what you can do for them, because often they don't know your area and they don't know that might be a simple solution out there you could do, and they just don't know. So that's the first part. You need to have a effective communication and know their language, and that is alone they'll probably take a fair bit of time to understand the jargon they use. And the other one is in making innovation outcome or the solutions, you probably need basically broaden yourself and think about what's the environment in the medical field. For example, Mark and I, we are looking at how to glue the polymer support with the donor's tissue. Now, you cannot just grow them in any environment where you normally do in the lab. You have to do them in the biological media. So the media have to support the cell to survive. And then when you do the gluing, which is essentially a chemical reaction, you grow them. And how you're going to do this reaction? This is going to be the reaction in the biological environment. You have to look at the toxicity, and looking at the damage, maybe the method you use. So the innovation comes to all sorts of these thinking from different aspects.
Thanks, Greg. Yeah, there's a lot of constraints you have to worry about working in a clinical environment, and I guess the eye is particularly sensitive, so lots of challenges for the bioengineering developments. Maybe, Mark, if I could come back to you, you've worked obviously with people in bioengineering for some time now. What sort of benefits and challenges do you find in working across the disciplines like that?
Well, I think Greg summed it up very well. The challenges early on are trying to communicate what the issue is and what each side can offer. The way I think about this sort of translational medicine is the clinician has a problem. They have the 1/4 of their patients they can't treat or prevent the disease. And they think of a sort of a half-baked solution. And then they take it to an engineer or a scientist who uses all of their skills and knowledge to give you a really a decent shot at a solution. Then you spend years and years working through that, making sure it's safe and effective. And eventually, once you've got something that really works, then you can take that back into patients. And so my role is at the sort of beginning and the end, and then chipping in with advice for with the engineers and scientists working on it in the lab. So I think the only thing I would add to what Greg's list of things that are difficult in this sort of collaboration is biology and the complexity of interactions between materials and cells is far too hard to predict, and things that should work don't work for some reason, and we eventually find out why. And some things, thank God, do work, and a lot of the things we do in surgery and in medicine just work, and we still haven't worked out exactly why and how. So I think that complexity of the hundreds of thousands of reactions that are going on inside every cell, and we're just tinkering with one or two things on the edges, it makes it very difficult to do it just from a theoretical point of view. You really have to try each of these ideas out.
Yeah, I've certainly had experience of that, and I can see you nodding there, Russell, as well. How do you go with predicting the performance of your implants?
Yeah, it is a real challenge. I mean, there's a tried and true process. We put a lot of effort into the design of our molecules. So, for example, we think about not only the process of release of the drug, but how the polymer will biodegrade. What are the final byproducts of that biodegradation process? What is the likely impact of that on the local environment, the tissue in the eye? Having then made your best effort to understand all that, as Mark says, then you get into biology, and we obviously use animal models to test our theories, and then you have a whole new level of learning again. And so there's an iteration to change the chemistry to then achieve something that you want. And then what you'll find is as you transition from those animal models into humans, of course, there are species differences that you have to address. And so for our glaucoma implant, where we were wanting biodegradation within 4 to 6 weeks of an end of a 6-month treatment period, we had to build a series of implants, and we studied them in rabbits, dogs, and ultimately now in humans, patients. But as we made that transition, the biodegradation period was different in each species. And so the reality is you just have to study it.
Yeah, it's really challenging. What's the longest sort of drug release period that you can achieve with your materials?
We can achieve up to 2 years, but the typical treatment period is 6 months. And the reason for that is not the chemistry or the technology. The reason is clinicians. The normal visitation cycle of a patient with a clinician is every 6 months. And, clinicians want to do things every 6 months, and they want to make sure the patient gets that 6 months worth of treatment. And they get a bit anxious if you offer things beyond that because, the normal visitation cycle is 6 months. So that seems to be the sweet spot for the longer, I don't know if Mark can comment on that, but that seems to be the sweet spot for the longer therapy products that we have under development.
Yeah, humans are even trickier to deal with than other animal models. But the problem is, I suppose, if some of these drugs aren't working well, or if there's side effects, you don't want them to go for 2 years without being able to change it. You want to be able to let that one wear off and try something else if it's not working. But, there are some of the drugs some of the new implants do last for 2 years, and that's obviously saves a lot of visits to the hospital and things. But if there's side effects, then you're sort of stuck with it and have to do an operation to try and remove the fragment if they really can't tolerate it.
Yeah.
Oh, very interesting. It's so much better than daily drops or monthly injections.
Yeah,
it's a vast improvement. Very exciting.
Yeah,
Mark, we have a. Sorry, go ahead.
So I was going to say it's even the drop burden. So our second asset is an implant to substitute for all the post-surgical care drops following, say, cataract surgery, for example, which can be significant. It can be 10 or more drops a day for a period of a few weeks. And so to be able to replace all that with a single implant is exciting as well. That's not so much the length of therapy, it's just the drop burden, the fact that you've got to put so many drops in your eye for that period of time.
Yeah, very attractive. Mark, we have a question from one of our registrants about new treatments or preventative options on the horizon for AMD. Can you comment on that?
Well, this is a little bit off topic, but I can give you some comments about macular degeneration. So with macular degeneration, we divide it up into a wet form where there's blood vessels that are growing through membranes and cracks under the retina. We've got a very good treatment for that at the moment, a monoclonal antibody, an anti-VEGF monoclonal antibody that we do have to give monthly or 2-monthly injections with at the moment. And so there's some talk about carrier molecules or maybe some of the products that Russell's been working on that give a slower release of that over time. The other form, which is sort of more than 90% of macular degeneration, is we call it the dry form. It's sort of a slow degenerative form. So I think that fits into that category where we don't have a good treatment at the moment. There's a lot of work being done in that area, though, with injectable drugs to modify the disease progression. There's some work using a laser that's been developed in Adelaide, a regenerative laser that can sometimes prevent, or it's thought to prevent the really serious complications from macular degeneration. And there's sort of, with these, the dry macular degeneration treatments, that again, there's drug delivery systems that are being set up that slowly release the drug over several years, which require a little surgical implant that people are putting in. So all those things are in trials at the moment, but none of them have, apart from the anti-VEGF monoclonal antibody, none of them are in practice as yet.
What's the sort of timeline required to go from, I guess, Russell, maybe one of your products through to being clinically and commercially available?
Yeah, so I can comment on that. It's a minimum of 10 years from the time you conceive and design your molecule through to the product being registered for sale and available for use by clinicians is a minimum 10-year journey, and oftentimes longer. Depends on the complexity. One of the challenges we have with our glaucoma product is because you're looking at such long treatment periods, the period it takes to demonstrate the product works in a clinical trial setting is longer than a conventional product. And so that extends your period of development and getting the product into the market. So it is a long road and requires people with deep pockets to support you. Yes.
Yeah, it's a significant commercial risk, isn't it?
Yeah.
Maybe I can add something to that. There was a paper written recently, or a couple of years ago, which looked at the lead times between invention and widespread use. And this was pre-COVID, okay? COVID just changed the rules. We can do it in 6 months now. That's no problem. Worldwide treatment. But everything else, so penicillin was about 40 years. This anti-VEGF drug I was talking about, incredibly effective, almost no side effects. That was invented in 1970. We gave the first injection in 2010. So, these are sort of blockbusters. Penicillin's a pretty good drug, almost no side effects. And before that, there was no treatment. And that still took 20 or 30 years to get into patients. So, and it needed a world war to really spur it on. So I think 10 years is really pushing the envelope, but maybe things will change now that with COVID, we've shown we can do the whole lot in 6 months, invent the drug in 2 weeks and do the trials and then just give it to 5 billion people.
Yeah. Though, Mark, I think people forget the fact there's 10 to 20 years worth of mRNA development behind the final 6 months. So it was still a 10 to 20 year journey, even for that rapid development.
Yeah, very exciting to see that last stage happen so quickly, but certainly we need all these technologies in the pipeline. And I guess in the tissue engineering field, I hate to say, but it's a longer pathway to clinical translation. Most often clinical trials are difficult and things like double-blinded trials and things are very hard to arrange. So tissue engineering adds another level of complexity, I think, in getting the required clinical confidence and regulatory approvals.
Yeah, I agree with that. I think cell therapies are particularly hard. There's one registered cell therapy treatment for the eye called Holoclar. It's a company in Italy, and they've gone through that whole regulatory process of taking cells from a patient, amplifying them in the lab, and then sending them back on a carrier material to be operated on. And that's the only one. That's the only cell therapy that I know about. That's it's a pretty basic form of tissue engineering that's actually commercialized at the moment. And that product has rewritten or has sort of set the framework ready for how these regulations and the regulatory authorities and the commercialization pathway for these types of treatments. So the EU are sort of busy writing the rules sort of to account for the way Holoclar sort of got it to market. So no one really knows how to do it. So every time we try and push it forward a little bit, we sort of are having discussions with regulators and which they haven't really got clear answers to us for.
Right. Yeah, it really is all developing simultaneously, isn't it? And I think bringing the regulators into the conversation early in the technology development is very important. So you have an idea of what their view will be, and sometimes it needs to evolve simultaneously, their rules along with our technologies. Yeah. We have another question from one of our registrants, and I'd just like to remind the audience, you can put your questions in the Q&A box, so please do add questions and we'll try to get to them as we go along. But our next question for the panel is, what would be the best way to enter this industry after graduating from a Master of Biomedical Engineering? So maybe Russell, would you be hiring biomedical engineers? What would you like to see them have in their kit bag to come to you?
Yeah, so I can share that we've sponsored PhD students in the past and encouraged young people to get into the industry. I guess the one common thing I recommend to them is to go, pre-COVID this was, to go overseas and work for a while. I think is one of the challenges we have in Australia is our geographical isolation. There's fantastic science done here, but it's not until you go and work overseas that you realise how good it is, and it gives you confidence that we are world class. And so I often say to our the postdoc to young scientists coming through is go and postdoc overseas, get that exposure experience. That network you'll build will be valuable for your entire career thereafter. And then come back with that set of skills. And I guess don't be too focused on a particular company or a particular area. When you're young, you should be like a sponge, trying to learn as much as you can. Yeah, I think that's the focus is for the first 10 years of your career, choose jobs that teach you how to do stuff and give you an opportunity to learn. That would be what I would say.
And would you think that it's common to need a PhD to come into working in this field, given how young and rapidly developing the technology is?
Look, it's not a requirement. I mean, we have both PhD and non-PhD qualified staff. Clearly, the PhD is highly valued in this industry. It does train you how to think, how to solve problems, you know, the principle of scientific method. All of that is really important stuff. And so if I look at our business, I would say it's more than half that are PhD qualified.
Right.
Yeah.
Yeah. And maybe Greg, you can comment on what it takes to get into a PhD.
Well, you need a good undergraduate study and outcome for start. But I want to add on what you just said, how you get into the industry. I just recently had a good example I want to share is that I had a PhD student in the last stage of the study, and he found this program called APR program, where that's the government funded the PhD student in the period of their study time for about 3 months to 4 months. They pay you to basically take a leave of absence, but all study away, and you do a different topic from what you do in your PhD. So this particular student, he was working on the polymer used for the electronic industry. And then he took this program, and I was saying our hydrogel can be worked as a porous support for making tissues, like a breast implant, for example. So we have a company, and the plastic surgeon is really interested in the project and want to have a little bit try on those. So I put him on the project for about 3 months, and he gets talked to the surgeon and goes to how the operations are done in the clinical places. He gets very excited, even he was not in the field, and he instantly created the interest in the field, and was very different from his PhD. And I think that's how you can make the link. And I would see in the future he can work in that part of the industry. So there are many ways, which in this pathway I didn't even know that. And clearly we have a lot of joint PhD project with the industry or with the medical field, but that's just a lot more of the options out there. Yeah.
Fantastic. I think that's a great advertisement for the value of a PhD. It really trains you in how to solve problems. And as Russell says, it sets you up to be a sponge and go off and do all sorts of different jobs. So we have another audience question now. Russell, I might direct this to you. How do you think we can gain the expertise to allow for medical manufacturing in Australia in a bigger way?
Yeah, look, we do have some medical manufacturing. CSL is a classic example of that. We've got the cochlear implant in Sydney as another example. So it's not that we don't have it. We don't have it in ophthalmology. To build it in ophthalmology, what we would have to do, I think, is one of two things. Either bring in the expertise from overseas. So we somehow encourage, I guess, entrepreneurs and experts in respective fields to build such a facility here. And it has to make good economic sense, right? So there has to be a driver as to why they do that. Or alternatively, Australians go and work overseas, learn those skills and bring them back. I think they're the two models that we would have to use. Yeah.
What do you think about getting the funding or the investment in Australia? It's very expensive to try and set that kind of manufacturing up. Is that a feasible thing for us to think that we can do?
I think it is. The funding will follow a business plan that has good economic sense. So if someone eventually has a product that it makes sense economically to manufacture in Australia, so they're the caveats, then the funding will follow that. It may need some government support to overcome some of the jurisdictional difficulties in Australia, like our geographic isolation and stuff like that. But yeah, I think it could happen, but you've got to have an economic business case that will drive it.
Yeah, and I guess there are particular products where it makes very strong sense, such as CAR T cell therapy manufacturing is starting in Melbourne because it makes sense rather than sending autologous cells halfway around the world. There's a strong driver to do that locally.
Yeah, and you know, the driver has to be one where geography is not so important. And typically you're looking at export industries, you're not looking at just local supply. And so, you know, I look at our product, it is possible. One of the advantages of our product, it's tiny. So our implant is, what, about 0.23 millimeters in diameter and no more than 2 millimeters in length. So you're dealing with a very, very small product. So shipping it overseas is not such a challenge as you would for some other products where weight becomes a prohibitive factor. I guess the other driver will be expertise. Is there specific expertise we have in Australia that can't be replicated overseas, and hence you have to manufacture here in Australia?
Right. Well, I guess as our research leads to new IP, perhaps that will drive new industry in that way. Yeah. Yep. Does the need to look at the export market mean that shelf life and stability of the product is even more important for us to consider?
I don't think it's more important, but it is important. So, you know, you really want to avoid well, as we've learnt with COVID, you want to avoid cold chain supply if you can. So the ideal product is room temperature. You want a couple of years shelf life. Most products are consumed within 12 months of manufacture anyway, but you want a minimum of 12 months shelf life. And if you can achieve that, then the export market is open to you.
Fantastic. Well, we have another question from our audience, this one from Ash Murphy. And Mark, I might direct this to you. How does a surgeon determine whether the handling properties of a material are suitable for the application? Do you have standardized tests, or do you just have a go, something in between?
Well, look, I'm interested. Ash, you sound like you're an engineer, because or either that or you've got a surgeon in the family. Because yeah, certainly when we first get the material, we have a go. We used to show a video where I'd grab the hydrogel and I'd stretch it a bit, and I'd sort of say, well, this is tough enough for me to do all this with, and then we'd have maybe an ex vivo model of surgery, and we'd sort of pull it through a few times and see if it tore apart. But collaborating with the engineers, they've got all these measurements for these things that I'd never heard of, you know, like Young's modulus and stress and strain. And so we can measure all of those things as well. We can sort of compare what a normal cornea is or a normal endothelial transplant is, and the stress and strain on those, and then put our materials through that. And so we can have a pretty good idea that they are very similar, you know, the handling materials are very similar. But sometimes the handling properties surprise you on the upside as well. The hydrogel that Greg's made, not only is it got all these is robust enough for surgery, but it has it manages to pop open inside the eye like a sort of a pop-up tent, which makes the surgery so much easier than the normal way of doing it, where the little bit of tissue that you're putting in is all scrolled up and can be very difficult to lay out flat without touching it. So we do a whole lot of standardized sort of engineering tests, but then we definitely have a go and tell him it's not good enough.
I've had many of those discussions.
I might add on that one is we do have the test method to have a quantitative values. But I remember, I'm not sure, Mark, you remember, before we were working on these, and I was working with O'Brien Institute, where Andrew, we were going to that meeting all the time, and we were making this porous hydrogel for breast implants. And I think one day we took the outcome and Mark across from the INE hospital to the St. Vincent, and was sitting our talk, and then found this material interested. And I know at that time he needs some support for the cornea. I remember very well, and we gave the material to Mark. Mark just put in a hand and just basically pushed it and said, "Well, this sounds or feel like similar to a human cornea. That should work." So that's why we try to so let's make a film, because we were making porous material then. We decided to make a film and try to do test. And that's how we actually start the project, and we are running for 10 years now. Yeah, so maybe initial feeling is very important.
Yeah, I've certainly presented materials to surgeons who do that sort of pulling and prodding and poking, and very quickly they'll give you feedback about whether they think it's something they would use. And I guess that's one of the key translation barriers potentially. If a surgeon doesn't want to use something, we may characterize it and it might have the right Young's modulus, but we also need to have the surgeons accepting the technology that we develop. So that's an important part of it. And I think those conversations as you do the development really help the research go faster and in a better direction. Mark, that leads me to another question I wanted to ask you about how ready do you think your clinical colleagues and patients and maybe funders are to accept this kind of new technology?
Well, look, we started off the one part of the technology, the Corgel project, is really as a material to help with the way we do corneal transplants at the moment. And we started that with an idea that really came out of the Translating Research at Melbourne program, where we rang a whole lot of corneal surgeons from around the world and said, you know, what is the unmet need in corneal surgery? What's the hardest bit of the operation? And would something like this be something that's attractive to you? And they nearly all said yes. You know, it's a part of the operation that drives us all nuts. And if it doesn't go well, that part, you're stressed and the patient doesn't do well, and sometimes the tissue is so damaged you have to do the operation again. So if you've got a big unmet demand like that, that makes the rate of application so much quicker. And so that's sort of been driving it on. Plus, we've got a business model. We've got that seems to be attractive. We've got a market in the US, and we've got a US company that's interested, an iBank over there that would like to sort of add this to their suite of products that they could then distribute to all of the surgeons in America. So I think having all those things set up, having a real unmet demand, and having someone who's sort of keen on distributing it for you, that really helps.
Yeah, that's hugely exciting. That sounds great. Another question from the audience, Greg, I might direct this one to you, relates to whether you really need a postdoc to enter biomedical manufacturing industry. So if you have a PhD, is it necessary to then go and get further research experience? What have you seen from your graduates?
Yeah, I think doing postdoc, it's definitely help you rather than it's not probably absolutely necessary, but it will definitely help you to broaden your expertise. And when we train a PhD student, they may be working on a specific topic, but they're really learning the research methods, where how we can identify the problem, find a solution for the problem, and basically writing your outcome as a thesis or publication for addressing that scientific question. Now, these skills, when you get trained, and you need to put it into the application. But a PhD project may not necessarily to direct to a specific application. So a postdoc probably experience will give you opportunity to look at the real application very often. And also, you never work in the exact topic when you get a job compared to the topic you did the PhD. PhD is not about you're going to do the same as you're doing PhD, then you do that for your job. It's about the learning for the method. Then you really need to broaden your expertise, perhaps with a postdoc for a year or two, maybe 3 years, and just really to see where you want to go. And after that, you probably want to find somewhere more permanent you want to work on. So I think postdoc position will help you rather than narrow you in your options in the future.
Yeah, I think in my experience too, it can be an enormous amount of fun. It really opens up your world. I went to the US for a postdoc and the experience was amazing because it did, as Russell said, it gave me a lot more self-confidence that actually my science was at that world level, but also the opportunity to interact with such a wide range of people. So it's a great thing to do if you can, before you maybe get a longer term job. Maybe Russell, what's your perspective in terms of hiring people? Do you prefer people to have more experience than just a PhD?
Yeah, that would be the preference. It's not always the way. It's not always the case. And, you know, in the last couple of years, it's harder because, you know, a lot of people can't leave Australia. But I can only reflect on my own experience. I also postdoced in the States for a couple of years. And the same experience that you had, Andrea, it gave me enormous confidence that I really was able to compete with what I thought was the creme de la creme at the time. And I think the other legacy feature from that experience was the network that I was able to establish over there, and which has continued even now, 20 odd years later. That was important as well. Yeah.
Yeah, I certainly found the same. I'm still in touch with the professor I worked with in my postdoc, and I have to say it looks really good on your CV. It boosts your competitiveness as you go on to the next things you want to do. Yeah. And we're drawing close to the end of our panel session, but if anyone has any last questions, do feel free to add them to our Q&A box. I can see Hala Renison has asked one. How much do you think patenting has helped you to further your career in this field or the field itself? And where could one hone such a mindset? So maybe, Greg, can I start with you? Your experience in patenting technologies, has that been helpful?
Yeah, in my group, we particularly involving PhD students in writing patents as well. And I think this is a very good training opportunity for them to see IP is important. And particularly when you work in the industry with a company like Russell's company, IP is very important. I found that almost very rare you will find a technology with no IP protection and people will invest on it because they just have to have their return. So they don't want to be spending all the money there. Suddenly everybody can make exactly the same thing. But IP training is very different from a normal PhD study training because you have to look at the importance of IP. Even the patent language is different. And what is important for the patent? What is the patentability? And those are very valuable when you actually train the student in the early stage when they're doing PhD. So when they go to the company, they will not get shocked and say, "Oh, that's so important. I didn't even know that," and that really helps them in the future. So it's important to involve them in those patent protection. I often, in my group, they're actually co-inventors for a lot of the technology. That also gives them the drive as well, because it's something I'm part of it, inventing this new technology.
Yeah, and can I comment on that? I think it's critically important. The reality is no investor is going to invest in a medical technology or a biotech company unless they can be assured they have market exclusivity around the product for a reasonable period of time to give them a return on their investment. And in our industry, the key to that is a defendable patent position. And I think that's an area we probably don't devote enough time and effort to. A patent is not a publication. In its simplest form, a patent is a contract between the community and the inventor, where the inventor discloses their invention for the community to use, and in return gets a period of market exclusivity in which to exploit that invention. So it's a contract between the community and the inventor. And so it's deliberately designed for an economic advantage. And I think people lose sight of that. I think a lot of them think, oh, they're just a publication, I can add it to my CV. No, it's not. And so when you think about it like that, it requires a different skill set to that you might get from your PhD or your postdoc. And it is even beyond what a patent attorney will prepare for you. A patent attorney is there to prosecute your patent application across the various jurisdictions around the world. But I think what we often lack is that expertise at the interface to really know how to use that invention for economic gain. That's the bit I think we often miss.
Yeah, I agree. I think that's really important and something that we need to have in our minds. And there's so many questions about timing and scope and how you frame those things that we really need expert assistance.
Yeah.
Now we have one more question coming through that we'll squeeze in, and it's how difficult is it to go from industry without a PhD into research? Maybe Greg, you could comment on this. What advice would you give someone who's interested in the field but hesitant about changing from industry? Do you think there's a pathway into research?
Yeah, I think so. I over the years, I see many people who were doing research very successfully without a PhD. And for example, a lot of graduates go to industry place where essentially they were working in the research center, and then they're doing very well over there. And often industry pays them to do PhD afterwards or during that time. It's still employed by the company. You look at the CSIO, there's two pathways in terms of people growing their career in CSR. One has a PhD, the other pathway has no PhD. Although the people has no PhD perhaps have a different task in the CSR structure, but they still can do a lot of major PhD work, research work over there. And I actually know one successful outcome over there is we collaborate. I know someone at CSIO is no PhD, but actually are in a very high senior position because of the success in the PhD. So it's not absolutely necessary. As I say, the PhD gives you very good training. It probably gives you a much faster way to go on to the PhD work. The research work. So you could think either way. You can be successful, but a PhD probably will help you.
Yeah, I agree. I know people similarly who've got into very senior roles in research without the PhD, if that's your choice. Look, we've come to almost the end of our time, so unfortunately we'll have to wrap up the questions there, but I would really like to thank everyone on behalf of the MedTech platform and the University of Melbourne, particularly our esteemed panel members for their contribution. So thank you very much, Mark, Russell and Greg, for your time and insights today.
Thank you, everyone.
And I'd like to thank you. I'd like to thank our illustrator, Zara. You've seen her illustration popping up, which is a fantastic representation of all the things that we've been talking about today. And I'd like to thank our audience for all your questions. We had a great discussion, I think, and your interest in these important issues. It's obviously a very exciting time in the field. We would appreciate your input on our post-event survey, which will be located at the bottom of this video stream and within the Q&A chat box. So we value your feedback to help us shape the next events, the future of our MedTech platform webinar series. So let us know what you'd like to see next. Thank you, everyone, and have a safe and nice day. Thank you.